Welcome to Delving into Academics, and for those who haven't heard of it yet, it's a podcast where we interview researchers from chemistry, biology and physics about what they're currently researching and also about their life as an academic. For this episode, Professor Alex Rowe will tell us a bit more about her research into treatments for malaria and what kinds of experiments she's doing to discover a treatment and possibly a cure for malaria. Okay, so I'm here with Professor Alex Rowe today. She's a professor of biological sciences and started out by getting a Bachelor of Medicine and Surgery from the University of Sheffield in 1989. She then went on to become a research assistant at a Papua New Guinea Institute of Medical Research, then on to the Ludwig Institute for Cancer Research as a research assistant as well. She went on to do her Doctor of Philosophy at the University of Oxford and on to the National Institutes of Health as a postdoc. She went back to University of Oxford for a year before coming to the University of Edinburgh and then staying here from 1998 till the present day. Thank you so much for being on this podcast. So, shall we jump straight in and ask, what are you currently researching?
Thank you, Jessie. So my research is all about malaria parasites, and what we do in my lab is we try to understand the interactions that occur between malaria parasites and human cells that can bring about very severe life-threatening disease in malaria. And this is something that remains a really huge problem globally. So about half the world's population are still at risk from catching malaria, mainly in the tropical, subtropical regions of the world. And unfortunately, malaria is still a very common cause of death. So something like about 500,000 people still die from it every year. And this is a treatable disease. So it's pretty shocking that this is still the state of things in the world. And what we're trying to do in my research is to understand exactly how these severe life-threatening infections happen and why they happen to some people and not others, and ultimately to see if we can design new treatments to help to prevent or to treat the most severe forms of human malaria.
Wow, that sounds so interesting. And as you said, malaria is still such a big disease and I guess a big problem in quite a lot of areas in the world. How do you study malaria and what kinds of experiments do you carry out to try and find cures for this disease?
So the parasite itself, it's a single-celled protozoan parasite, and we can study it in lots of different ways. Most of our work we actually do here in Edinburgh in the lab, so we can grow the parasites in cell culture. So this involves having flasks in which we have medium with lots of nutrients, glucose, salts, vitamins, all kinds of things that the parasites need to grow, and also red cells, because the parasite, the form in which we study it, it lives inside red blood cells. So we have to add red cells to our cultures for the parasites to invade into and to help them grow and develop. And we also have human serum, so blood components that the parasites also need. So here in Edinburgh, we're growing the parasites in the lab. We're also growing some of the human cells that the parasites can interact with that leads to disease in humans. And one of the real hallmarks of life-threatening malaria are interactions that occur between the parasite and cells called endothelial cells, which line the walls of blood vessels, particularly small blood vessels in the brain and in other key organs in the body like kidneys, liver, spleen, and so on. And what happens is that the infected red cells that have parasites inside them can become sticky, and they adhere to the walls of the blood vessels, and they cause blockages to blood flow. And this is one of the ways in which the parasites actually cause damage to the people that they're infecting. So here in Edinburgh, we try to study these interactions by growing the parasites, by growing the endothelial cells that line the blood vessels, and we can do experiments where we mix the two together, and we can then study their interactions by looking down a microscope, for example, by studying factors that might block this interaction, or looking for drugs or antibodies or other reagents that could maybe reverse these interactions. And what we'd really like to do, our long-term aim, is to come up with a therapy that can reverse the ability of the parasites to stick in the blood vessels, and this would then potentially free up the blood flow and restore people to health quickly and stop them from dying from severe malaria. So we also have other ways of studying the interaction. So there's the work that we do in the lab. We're also involved in some work where we actually give malaria to human volunteers, and this is done with some collaborators working down in Oxford, and we're involved in studies where people get infected with the parasites and then they're followed over about 1 to 2 weeks. We're able to study how the human body responds to the infection and how the immune system in a person is able to recognize the parasites and mount a response to the parasite, and equally how the parasites respond to being in a human body and whether there's much variation in the types of genes the parasites are expressing in relation to the type of immune response that the host, the volunteer, is mounting to try and kill the parasites. So there's always this battle going on, I guess, between the parasites trying to survive and transmit and spread the infection to other people, and the infected individual whose immune system is trying to detect and to kill the parasite. And a lot of the factors that affect the outcome of the disease are all to do with this battle between the parasite and the host.
Yeah, it sounds quite exciting when you put it in like terms of a battle, and it is such like the parasite does want to survive, and then the human will have this response, and it possibly might vary human to human. So do you find that there is a difference to studying it when you put malaria in humans to when you're studying it in the lab? How close are your lab experiments to real life scenarios?
That's a really good question, and it's something we always worry about with our lab experiments. They're obviously greatly simplified, which in a way is a good thing because it allows us to pinpoint specific factors that might influence some of these interactions more precisely because we're working in a simplified system. But the downside is it's a worry that we're missing essential components that happen in a living, breathing person that you don't get when you're just studying a cell line in a dish, essentially. So we always have to be very aware from our in vitro lab experiments that we can learn really important things, but we have to be sure that they are actually relevant to malaria in real people, which is one reason why we also do the work in human volunteers to go along with the lab experiments. It also involves taking into account other factors, looking at things like genetic variability in real human beings that we don't normally come into play in our lab experiments. We're normally using a cell line, and we might just use the same cell line for a whole set of experiments, whereas with people we're obviously using a different individual every time you infect a volunteer. Or the other thing we do is to collect parasite samples from naturally infected patients. So some of my students are involved in work in countries where there is lots of malaria still, unfortunately. So we collect samples from hospitals where children come in with malaria and life-threatening malaria, and then we study the parasites from natural infections, so we can compare some of the factors that we find in our lab experiments with these real world parasites and patients to try and look at the relevance of our lab experiments in the real world. So that's certainly a very important thing to always think about with the research that we do, is how relevant it is to the real world. I think I also touched on there this idea of genetic variability, which is something that applies to both the parasite and the host. But on the host side of things, we know that malaria has been a massive selective force on the human population, and there are lots of inherited genes that have become very common in malaria endemic countries because they actually protect people against malaria. And one of the most well-known ones that people often learn about in biology at school would be sickle cell trait. And this is a mutation in hemoglobin inside our red cells, which helps to carry the oxygen around in the blood. And in sickle cell trait, there's a single amino acid is changed, and it causes the red cells to adopt this unusual sickle shape, hence the name. And for people who inherit this polymorphism of their red cells, if they just get one copy, then they're protected against malaria. If they get 2 copies of this gene, then unfortunately they have a very serious disease called sickle cell anemia, which can be life-threatening. But for the people who only inherit 1 copy of the gene, then they're protected against malaria. And this is something which has become very common in many parts of sub-Saharan Africa, for example. And that's the kind of textbook example. But over the past few years, one of the other things we've done in my group, one of our areas of focus, is looking at other human gene polymorphisms that influence the host-parasite interaction in malaria and actually protect people against malaria. So we've identified some other genes that have this similar effect to sickle cell trait. For example, your blood group affects your risk of malaria. So most people are familiar with the ABO blood group, and for people who are blood donors, they may well know their blood type. They'll either be A or B or O or AB. And within these blood types, one of the things we've discovered is that if you're group O, then you're protected to some extent against malaria. So it's not all or nothing, but it means that you're less likely to develop severe malaria if you get infected if you're group O. So that's another example of the type of work that we've done in the lab here.
Yeah, it's so interesting that genetics plays such a seemingly important role in how susceptible you are to malaria. What makes the sickle cell trait so protective against malaria? How come patients or people who have this trait won't get it? What makes it so much stronger or resistant?
So the sickle cell trait in particular is something that many people around the world have studied for the past 10 or 20 years to try and understand exactly how it protects people against malaria. And it seems to work in several ways actually. There's some evidence that the parasites just don't grow quite so well inside red cells of people who have sickle cell trait, and this may be a part of it, but it's probably not the whole story. And the other part of the story seems to be that having this mutation in the red cells actually affects the ability of the parasites to become sticky and to stick to the walls of the blood vessels. So going back to what I was telling you at the start, it seems to be that if you have sickle cell trait, then it influences the ability of the parasites to make these interactions with the host cells that block the flow of blood and end up causing some of the most life-threatening forms of malaria. So if you have sickle cell trait, that's less likely to happen. And that's also true for some of the other red blood cell polymorphisms that protect people. So again, the blood group O that we talked about, that affects an interaction between the parasites, the infected red cells, and uninfected red cells. So you get these clusters of cells forming called rosettes, and the rosettes contribute to blocking flow of blood in the blood vessels. And for people who are blood group O, the rosettes are smaller and weaker and more easily disrupted. So the flow of blood isn't compromised in the same way if you're blood group O. So that's reducing your risk. So it's one of the reasons why exploring the genetic factors that protect humans is of interest, because if we understand the mechanism of protection, then we can start to think about maybe designing therapies or looking for drugs that would have the same effect as the polymorphism that naturally protects people. We can try and mimic that with some kind of a treatment to help reduce the risks of severe malaria.
Okay, and from the studies you've done so far, what types of therapies and drugs do you think will, or how do you develop therapies and drugs from looking at the genetics and then applying it to malaria infected patients?
That's a very good question, and it's a very difficult thing to do, unfortunately. You know, if it was easy, we or other groups who work in this area around the world, we would have done it by now, and we would have saved lots of lives, and we'd all be feeling, you know, very pleased with ourselves. Sadly, even though the amount we've learned about how malaria parasites interact with human cells has really grown over the past 10 or 20 years, there's no example yet of that new knowledge actually being translated into a new treatment. People are trying to do it, and people have come up with lots of ideas about how to do it. There are lots of difficulties, which include the fact that the parasites are so variable genetically, and for example in my lab, one of the things we've done is to identify the proteins that parasites make that make the infected red cells sticky and then cause them to block up the blood flow in blood vessels. And we were hoping that by identifying these parasite molecules, we could maybe think about designing a vaccine in which we could immunize people in malarious countries so they would have antibodies in their bloodstream that would prevent the stickiness from happening. And this seems like a logical approach, but the problem is that the parasite proteins that actually mediate this sticky interactions are highly variable. So you would need a vaccine that was basically a cocktail of many thousands of different types of parasite protein in order to be able to induce antibodies that recognize every possible parasite version that existed out in the real world. So that's proven to be a very difficult thing to do because of the variation in the parasite. We do know that people naturally become immune to severe malaria after a small-ish number of infections, maybe after something like 5 to 10 infections, people do become naturally immune to severe disease, and many people are trying to understand exactly how that process works so that, again, it could be mimicked. But as yet, no one fully understands how that works, and whether that involves antibodies against these sticky proteins or not remains a controversial topic. And it might do, but it also might not, and there isn't very strong evidence either way. So to come back to drugs or other approaches, there have been some potential drug treatments that have aimed to reduce these interactions that block blood flow, and they've mostly come about just by people doing empirical experiments, just thinking, oh, let's try this, that and the other, let's just put it into our assays and test and see if these things disrupt the interactions. And some candidate therapies have been described, but unfortunately most of them have had side effects that have meant that they couldn't be used in patients. In particular, there's been a class of drugs that we've worked on and some other groups around the world have worked on, which do disrupt these interactions, but they have the side effect of causing bleeding. And obviously you wouldn't want to give a treatment that might cause bleeding to a child who was already very sick with malaria in a setting where there may not be the resources available to give safe blood transfusions and so on, for example, if bleeding did happen. So It's challenging to come up with a therapy, and it's even more challenging to come up with a safe therapy that you would be happy to give in a resource limited setting, and where you don't need all of the paraphernalia of a intensive care unit and all the backup facilities, which unfortunately are just not available in many of the parts of the world where malaria remains such a big problem. So that is still a difficult thing to do. We and others are continually exploring new avenues, and at the moment, for example, we're looking at some antibody fragments as a possible therapeutic avenue that maybe can disrupt some of these interactions between the parasite and the host cell, and we're exploring that at the moment. The other problem we have is that it's actually very difficult to test therapies for malaria, and for severe malaria in particular. We've talked about the types of experiments we do, and I've told you how we do experiments in the lab, or we sometimes do experiments in human volunteers. We don't really have a very good animal model that can be used to test out new therapies, and this is a real problem in malaria. So the parasite that causes the most deadly form of human malaria really only infects humans and a few higher primates. So we don't have a good mouse model, for example, to test potential therapies on, and that is something which is difficult because it limits. You can't test very large numbers of potential therapies in human volunteers, for example. You can only do very small, select numbers of experiments if you're actually needing to involve humans and infect volunteers. So it's an issue that's a problem for the field in general, the fact that we don't have a good animal model to use. And there are similar problems in some other infectious diseases as well, things like tuberculosis, for example. The animal models are difficult and limited and not always looking quite like the human disease.
Yeah, it sounds very difficult. There's quite a lot of challenges to come up with a therapy or a cure to malaria. It's actually quite interesting. Well, I say interesting, but it's, I guess, intriguing, the genetic variation of parasites. What kind of genetic variations are you seeing, and how do they present themselves in the parasites in the way that they infect humans?
So these parasite proteins that we have been focusing on, particularly, they're part of a large family that are encoded by a multi-gene family, and they do something that's seen in actually a few different types of parasitic infection, and also similarly in some bacterial infections. They do something that's called antigenic variation, and this is something that I think some biology students may have come across already, but it's a process whereby a pathogen on its surface has a particular molecule, and within its genome it encodes lots of different versions of that molecule, but only one of them is put out on the surface of the pathogen at a time. And what happens is, when the host immune response starts to recognize that this is a parasite and it's going to start making antibodies and reactions against the parasite, it makes antibodies against the version that's on the surface, and after, say, a week or two, the host might be able to kill those parasites that have this first version of the protein on the surface. But the parasite has a real trick up its sleeve, which is that it can switch to then express a different version of that protein. So if you think of it, I guess, as like wearing a cardigan that's being recognized by something. You start off wearing a red cardigan, but once the enemy can detect the red cardigan, then you take that off and you put on a yellow one, and then you can't be seen anymore by the enemy because it's a different coat. And then once they can recognize the yellow one, you take that off and you put a blue one on, and so on and so forth. And this is a way in which parasites or other pathogens can prolong their infection in a host, is by constantly changing their surface coat. so that they can't be recognised by the immune system of the host. And this allows parasites to persist for months or even years in some cases in people who are infected. And for malaria, this is important because in parts of the world where malaria is very common, the disease is very seasonal. So you probably know, most people know, malaria is transmitted by mosquito taking a blood meal. The parasite then develops in the mosquito, and when an infected mosquito then goes on and bites another person, they can inject the parasites into the blood of that person. So you need mosquitoes to be around to spread the disease. In some malarious countries, the mosquitoes are only around for maybe 2 or 3 months of the year, and it's related to when the rainy season happens to provide breeding sites for the mosquitoes. So in this case, what happens is that the malaria season might only happen for 2 or 3 months of the year, and then the remaining 9 months of the year there's no malaria transmission at all. Obviously, from the parasite's point of view, they have to be able to survive for 9 months during the year without being spread, without being transmitted. Otherwise they would just die out because there are no mosquitoes around to transmit them from person to person. So this ability to persist within the host, to keep changing their surface coat, is essential for the parasites to be able to survive in many parts of the world. So it's an important part of their biology. And in malaria parasites, it's this same family of proteins that do this antigenic variation are the same ones that are the adhesion molecules that do this sticking to the host cells to block the flow of blood. So this particular family of proteins is of major interest, and we study them, many people around the world study them, but they are very variable in their sequence. Each parasite isolate has about 60 different versions of these proteins, and every isolate that you collect from a different patient has a different version of 60. So the diversity in this gene family overall in the real world is just enormous, which is what makes it so difficult to target it in terms of a drug or a vaccine, so on. So we have a big challenge.
Yeah, it definitely sounds like it's the amount of variation that there must be in these different samples and in real life scenarios sounds immense. I guess I didn't quite realize how variant malaria could be, and I could see how it could make it such a difficult challenge, because you can't attack it in just one way. You'll have to think of multiple ways or have multiple antibodies in order to kind of cure it or fight back. So yeah, that's quite interesting, really.
Yeah, although as I say, it is the case that in the real world people do become immune to severe malaria after being exposed for a few years. In sub-Saharan Africa, which is where most of the deaths from malaria occur, the severe disease is really only seen in children under the age of about 5. By the time children get a bit older than that, they've become immune to the most severe forms of the disease. They can still get infected, but they don't get so sick. So we know that it's possible to develop resistance, immunity, tolerance. We don't fully understand quite how it works, and if we did understand exactly how that worked in natural infections, then it might be easier to design therapeutics to mimic the effect. But it's a big problem still, and it's a big challenge to overcome.
Yeah, absolutely. And with your current research, you mentioned studying some of the factors of the parasites in these types of systems, and I was just wondering, what kind of factors are you researching or looking into?
So mostly we're studying this family of proteins that I was telling you about that undergo antigenic variation in malaria. We're characterizing the sequence of the proteins and working out which parts of the proteins are involved in adhesion to host cells, and we're looking to see if any parts of those proteins are conserved across different parasite isolates that have the same function and to see if we can pull out conserved parts that we could think about using in a vaccine, for example. So that's one example. And the other thing we're doing now is we're using a new technology that you may have heard of called CRISPR-Cas9 genome editing to modify the parasites, to change different sequences and to see how that affects their binding to host cells, for example. We've only started doing that in the past year or so, and it's giving us a whole range of new approaches and new tools that we can use to study exactly how the parasite is binding to host cells, to really understand it at the kind of detailed level that you need in order to be able to really start trying to rationally come up with ways of blocking these interactions.
Yeah, and it sounds quite exciting that you mentioned using CRISPR gene editing, but only very recently. Do you think this is something that could potentially in the future lead to us knowing more and more about this parasite and maybe finding the cure to malaria?
I hope that will be the case. It's certainly been a problem over the past decade or so that it was very difficult to do any kind of genetic manipulation of malaria parasites. I mean, it was possible, but it was difficult. It's very slow and laborious to try to do any kind of genetic manipulation by traditional techniques. But now that CRISPR is here, it's so much more efficient. It's just, I think, opening up a whole range of avenues that people can begin to explore that just weren't possible 5 years ago. So yes, I am optimistic that it's really going to make a big difference. I guess the other thing to say is that that the types of therapy that we are trying to develop is all to do with treating the severe life-threatening forms of malaria. There are many other researchers who are trying to come up with new types of therapies that will just kill the parasites, that can be taken as tablets before people get severe disease. And many of the existing medications that are out there that kill malaria parasites are actually really effective. You can cure malaria if you get the right treatment and you get it quickly enough. The problems still come in areas even where there are good anti-malarial drugs available. If people don't get treated fast enough, then they can develop infections that go into this severe life-threatening form, where they end up in hospital with life-threatening disease and needing some extra therapies. And this is the part of malaria that has really lagged behind everything. There have been new anti-malarial drugs over the past 10 years or so, but there have been no new treatments for the most severe life-threatening forms of disease, where you would give an anti-malarial drug to kill the parasites, but you also need to give other types of therapy to try and actually address or reverse the pathology that's underlying the severe symptoms, which may be things like coma or difficulty breathing or being very anemic, all of these things. You can give a blood transfusion for anemia if there's blood available, but the other features of severe malaria, like being unconscious and difficulty breathing, there's really no effective treatments at the moment. So it's an area of the disease that really needs progress to be made.
Yeah, absolutely. And it sounds like with all the challenges that you're facing in combating the severe cases, that more people should probably be looking at severe malaria and then working on how to kind of combat its effects in its entirety. And so where do you see this area of severe malaria research going in the future and progressing to? What are the kind of next steps to research in this area?
I think that there's still a lot more to be learned about the genetic diversity of the parasite that we've talked about to some extent already. We need to know a lot more about that in future, and I guess as well as things like CRISPR, the fact that there's so many new genome related sequencing technologies have developed enormously over the past 10 years or so, and they're making a big difference as well, because you can begin to look at diversity in parasite genomes on a scale nowadays which was impossible to imagine 10 or 20 years ago. So I think that will make a big difference. A lot of people who work in the malaria field are trying to work towards eliminating malaria in some countries, trying to stop transmission altogether, and a lot of funding goes into those areas. And just to give people simple tools like bed nets and better access to drugs and things like this, which can actually make a really big difference. And all of those things are hugely important, but obviously it's also really important that the basic science research keeps developing at the same time. And even if cases of malaria do go down, and in some countries they've been controlled quite effectively with some of the existing tools, but unless malaria is eradicated completely, which is really not very likely, then there's always the risk that it's going to come back again, and there's always the risk of small numbers of people getting severe disease. So it certainly needs to be part of the research agenda going forward, along with better anti-malarial drugs, better public health tools. All of these things need to progress side by side, and hopefully will do in the future.
Yeah, absolutely. It sounds like such a interesting field with quite a real world impact on people's lives, on countries that may not have, you mentioned, may not have the resources that we have in the UK, and I guess the dream or the ideal world would be to eradicate malaria, but as you said, this, and as you have described, it sounds quite difficult, but it sounds very optimistic that in the future there'll be lots more ways to maybe prevent it getting to the severe cases or even transmitting to other people. So yeah, it sounds a field that people should get into a bit more. So you sound very passionate about malaria and your research. What is it about studying malaria and the parasites that really intrigues you and wants you to learn more about this?
I initially trained as a doctor, and it was while I was a medical student that I first became interested in malaria, and I was fortunate enough to spend an elective period abroad. So I worked in Papua New Guinea at a research institute, and that was my first real exposure to malaria research, and I just became completely fascinated. I was hooked by it, and in fact I even took a year off in the end during medical school and went back to Papua New Guinea so I could do some more research. So I got into it so much during my brief first visit, which was just a couple of months. I then followed that up with a year off so that I could do more research, and that was really when I decided that this was what I wanted to do with my life. I wanted to understand this and do some research, and I suppose try to make a difference. And I think that's true for many people who work in malaria research. It's one of the big global health problems, and it's crying out for new approaches to try to make the situation better. And so I think that is a big motivating force for me and for many people in the field, is the thought that whether you in your own personal research manage to make a new therapy or a vaccine, that, as we've talked about, is a big challenge. But even if that doesn't happen, the hope that the research that you do might help someone else to do that, or the people that you train in your lab will go forward and contribute to the area in the future as well. So I like to think of science as a big team effort, and that we're all doing what we can to contribute to the team approach. And at the end of the day, there will be a vaccine that works against malaria. I think there are some potential vaccines in various trials of various sorts at the moment which don't have great effects. They're not really very good, but for the future going forward, hopefully there will be advances, and lots of people will be involved in future successes, hopefully.
Yeah, and I love the idea of that science is just this one big team, and if you don't get it, maybe someone else will, and another team will come in and they'll be able to find this vaccine, or maybe even build from your own research and what your team has been able to produce. So yeah, that's an absolute lovely reason to be researching malaria. And you kind of picked up on this big team idea and how your research will help or could factor into other things. And has malaria research and research into parasites, maybe even in your techniques, been able to contribute to other areas in infectious diseases or to other teams elsewhere?
Ooh, that's a good question. I think there's no doubt that some of the research and training that goes on within the malaria field, particularly, nowadays there are lots of great collaborations between labs in the UK, in Europe, in the States, in Australia, around the world, and in countries where there's lots of malaria, so sub-Saharan Africa, parts of Asia, Papua New Guinea we've mentioned, and so on. There's lots of great collaborations which benefit both sides of the interaction. So you have scientists, for example, from Edinburgh going to work in Uganda and learning about what malaria is in the real world, and then hopefully the reverse, scientists from Uganda getting the chance to come to work in Edinburgh to learn new techniques and receive training, so that there's lots of benefits from these interactions which don't necessarily just apply to malaria, and that training, capacity building and so on that goes on, I think, can benefit research generally, not only malaria, but then get applied to other medical problems in countries of interest. Whether there are any specific examples of, I don't think I can think of any specific examples of research in malaria definitely having a benefit on other diseases.
Yeah, I mean, it sounds like it has quite a big impact on the training side, I guess, so especially when you do have these sorts of collaborations, and the amount that you can learn from doing this on both sides will be quite informative, and then also be able to help maybe develop your knowledge on malaria or other diseases, or techniques on how maybe you cure people or go about finding cures for diseases. It sounds like it does have quite a big impact already without even the actual research itself. And so, I mean, going back to research as a whole, what is your favorite thing about being a researcher? I know you kind of touched on why you like malaria probably specifically, but what about being a researcher and an academic really intrigues you and keeps your interest going after so many years of doing it?
I think the thing I love about science is that you're always learning something new. You have to keep learning, you have to try to keep an open mind, you have to try not to become too blinkered or dogmatic in your approaches. You have to, I think, try to maintain a degree of humility in terms of your own ignorance, and try to remain open to new ideas. So I think it's just the fact that it gives you the opportunity, the necessity, I guess, also of just constantly learning new things. I think we're very privileged to be able to have a career in which learning and being open-minded and trying to make progress in this way is a large part of what we do. So yeah, for me that's the best part of being a scientist.
Yeah, and that's also why I'm in science. The continuous learning is what interests quite a lot of people in science as well, because science is developing. I mean, just from our talk in malaria research and all the developments going in there, as you said, like 10 years ago there were things within your area that just weren't really thought of, and now they are, and it's almost like you can adapt, you can discover new things, and yeah, there is definitely a great interest in that. And so with looking at undergraduates then going into maybe research or just kind of looking towards the future and how they can progress and such, what kind of advice would you give them? Maybe if they don't know what they want to do, they're kind of still stuck, but they do like biology, or whether they know they want to go into research and continue on there.
I would encourage people to read around, I guess, to explore different possibilities. If they are interested in research and they're undergraduates, I'd really strongly encourage them to try and do a summer internship of some sort, try to get some research experience. There's nothing quite like being in the lab. If it's wet lab science that you're interested in, there's nothing quite like being in the lab to really get a kick out of doing your own experiments and really understanding what's involved. Or obviously some people don't want to do wet lab science, but they want to do computational biology or other aspects of biology. I think find what you're interested in and try and get some experience doing it is what I would recommend to people. And I guess explore lots of different areas through your reading or listening to podcasts or watching YouTube or whatever, all the different ways in which you can access science nowadays. Look at Twitter, all of these things.
Yeah, that's some really great advice, because there's so much out there where you can learn about different areas of the science or the subject that you're studying, and then go from there, because there might be an area that you've never heard of through lessons or through your courses, and you're like, oh my god, this is amazing, like you experienced with Papua New Guinea and malaria. So yeah, that's really wise advice. And I mean, speaking of reading around your subject, unfortunately we have come to the final question of the podcast. As much as it has been absolutely lovely to speak with you and to have this talk about your research, but what is an inspirational book or article that you've read that you would really like to recommend to our listeners to go and read themselves?
So I'm a massive fan of a science writer called Ben Goldacre. He's actually a physician by training, and he's published I think at least 2 or 3 books now, and there's one called Bad Science and there's one called Bad Pharma, I think, and he wrote a column in The Guardian for a while as well, and he basically writes about dodgy science and how to make science better. And I think there's lots of great science being done nowadays, but there's also a bit of a problem with some science not quite being up to standard, and something called the reproducibility crisis in biology, that it's become apparent that many high profile studies can't be reproduced by other people. So there's this real need for stringency and high standard and reproducibility. And Ben Goldacre writes in a very entertaining way about examples of poorly done science and how to do it better. So I would really recommend to students to read some of his books or articles. They're really intelligent, they're full of good advice, and they're funny in many cases as well. It's wonderful stuff, so I would recommend people to read some of his books.
Yeah, and that's such a great suggestion and recommendation, because that's the sort of thing that will make you a good scientist for the future, or even for your undergraduate or postgraduate studies and research as well. So that's absolutely fantastic recommendation. Thank you so much for being on this podcast. It's been absolutely fantastic to have you and to discuss more about your research, and I hope it continues to develop and you find a cure or treatments for malaria in the future.
Thank you very much.
So thank you for listening to this week's episode of Delving into Academics, and I hope you found it interesting. Please like, review, comment and subscribe wherever you're listening to this. And if you want to find out more about the researcher, I will have their university website page linked in the description. See you in 2 weeks for another episode.